Thank you for coming today. This is the part of the Genomics for Teachers series, and I'm very delighted to introduce Dr. Crawford, who is here from a lab on campus, the Sarah Tishkoff's lab, who works on genomic variation. She and her lab members look at human evolution and disease risk in humans. And so Dr. Crawford actually before coming to Penn, he was at the California Academy of Sciences. And so today he will be talking about evolutionary genomics. And I also didn't introduce myself. So I'm Kristen Field with the Nanobiointerface Center. And we're delighted to have you here. I think everybody signed in, but if you didn't or need to fill out something on the way out, please do your checkout time if you haven't done that already. I think everybody got a handout, but if not, there are extras if you want to take one when you leave. And if you want, we can make the PDF of the slides available by email, if that's helpful. So without further ado,
Dr.
Crawford.
Thanks, Kristen. Is Kristen right?
Kristen is.
Good. All right. So I'm pretty informal. I know it looks all formal here, but you guys should interrupt me. I know you're high school teachers, so you're probably good at that. That's the reputation. All right, so now that I've insulted you, then you're really going to come down on me. And you have like the slides that you can read ahead. All right, so this is probably a bit of an overstatement, but to keep it simple, I sort of start off with that there's sort of two main questions in evolutionary biology that people are really interested in. And you could probably like make 5, but to talk about things I know a lot about, and to keep the topic sort of like with some bracketing around it, I kept it to 2. And they are, what is the history of life? So this is lots of biologists and evolutionary biologists are really interested in delineating relationships among species or families, like higher level orders, or between populations. And so there's a lot of work that goes into just figuring out the relationships among species, or we say taxa, so anything up on the tip of a phylogeny, like a tree here would be a taxon or a taxa. And the other question that's getting a lot of work these days is understanding how adaptations evolve, and also sort of how new species evolve at the context of the genome. So like how much divergence in the genome do you need to make a new species? So I'll talk about that a bit as well. And I sort of want to start off with, before we had DNA sequences, there was tons of work done on morphological data. So this is like looking at differences in bones, or it would be like paleontology, which is still the only way to study many extinct animals because that's all you've got as a bone. Or people look at organ shapes and just study things like developmental biology, looking at how the fetus develops in a whole bunch of different species, and which is great, except for this one big problem, and this is this concept called homoplasy. And the basic idea, and the reason I have a bat up here, is that if you compared a bat and a bird, and you said, well, they both have wings, then you'd think they're the same, sister to each other. When of course that's not the case. That bats independently evolved wings from birds. So this, that's like an easy system to figure it out. But when you're looking at like some little flange on some scapula or something, and in a bat and in a mouse, is it the same little flange or is it something different? So that's when the homoplasy comes into play, and people make mistakes, and then they get the wrong phylogenetic associations, and the tree is incorrect. So back when you had just morphological data to do these sorts of studies, you didn't know if you were right or wrong. You thought you might be right. Most of the little traits you looked at support one arrangement, but there are a few that are discordant, and there wasn't a good way to work with that until people started sequencing DNA. And the nice thing about using DNA sequences to figure out the relationships among animals is you can actually model how evolution works mathematically. And I'm not going to go into that at all, so don't worry. Except to say that like the simplest way you could do it is there's 4 nucleotides. They have an equal chance of one mutation. The model would be that there's 4 nucleotides, and your chance of mutating from an A to a T, or an A to a C, or A to a G is the same. And of course, you could get more complex than that and talk about the position in the gene and how that changes the model. But that'd be the simple way of doing it. So you'd take DNA sequences of the same gene from many different species, line them up, model the evolution, and then you could infer a tree from that. And So like a simple case or a simple approach to doing this is something you could potentially do with high school students, and that's what I was told to present with sort of high school students in mind, is a parsimony tree. And parsimony is just, like parsimony or Occam's razor is that concept that the simplest solution is the most likely correct one. So if you started with, if your final species are represented by these 4 little sequences of 3 nucleotides, so AAA, AGA, GGA, and AAG, you could have a phylogeny that shows that the ancestor was all A's, and then it split, and they got a G here, and then they got one more G. And on this other branch over here, it could retain the ancestral sequence, and then there was one mutation to a G. And that returns the AA, AAG, GGA, and AGA, and with 3 mutations necessary to produce that. And this other possibility, you keep the same ancestral sequence all the way through, but then it requires 1, 2, 3, 4 changes. So the parsimony, if you're using a parsimony criterion to determine what the right tree is, you'd say this is more likely to be right because it only has 3 changes. So you could, if you're giving like a test question, you could expand this to whole, 4 or 5 different possibilities, and then ask your students to pick which one is the simplest solution. And you could switch this out. It doesn't have to be, nucleotides. It could be, beaks or claws or whatever the trait you want it to be. So people use this for, morphological traits as well as for sequences. Does that all make sense? It's probably the most complicated. Well, maybe not, but anyway. So this sort of type of study, where you sequence a number of individuals, or pick your favorite group of animals, sequence a couple genes, and then use software, because nobody does this by hand anymore. It's too hard to make a phylogeny. There's a whole cottage industry of this sort of type of study starting probably in the late 70s until the present. And people have used this to resolve all sorts of questions, like figuring out who the closest relative of humans is, which is the chimpanzee. But I think there was some question about that up until there were DNA sequences available. They identified a whole new kingdom of animals. I'll talk about that more in a second. The Archaea, the position of snakes within lizards. So as my background's actually in herpetology, studying lizards, so that's a good example. And many more. So people study beetles, and they sequence genes from beetles, and they sequence genes from orchids, and all sorts of anything you can get DNA out of. You can, take this approach with a couple of genes. So this Archaea example is a good one because back in 1977, I think his name is Carl Woese or Wose, and George Fox sequenced some RNA genes from extremophiles. I think they got stuff from what's the national park? Not Yosemite. Yellowstone. Thank you. But it might have been some other location. But they're cool because it turns out that the individuals or the animals they sequence are actually more closely related to. Well, they're not. They look like they're not. They look like, if you looked at them like on a microscope, they look like they're prokaryotes, like bacteria. They actually are a little bit more closely related to. It's okay. To eukaryotes. All right. So I'll say that again. So these are single-celled organisms, live in extreme environments like hot springs. You would think, based on their morphology, that there may be bacteria, but they actually are their own little group that's more closely related to eukaryotes. And that was back in 1977. And then it turns out that actually the relationship of snakes with lizards was something people wanted to know. A postdoc in the lab I was in as a master's student did research on that. And this is sort of more of an exemplar. Like, there's tons and tons of studies out there that do this sort of thing with, you name it. If people can get DNA from it, they've probably started to look at the relationships among the taxa. So the snakes fill out here. It actually turns out it's not totally resolved where they fall. And the other cool thing is this weird fossorial, which means like lives in the underground, lizard from central Mexico is the ancestor, or his sister, I don't like to say most, looked like his sister to all other extant lizards. That was totally unexpected. But any questions so far? No? Okay. And then people get fancy now, they can go online and you can download. Every time you publish a paper, you have to put your data online so people can go find thousands and thousands of sequences from like 2,800 species of amphibians. This is actually a phylogeny. It's just wrapped around in a circle. So all these little, there's like many, many, many tips. And these are all frogs here, and there's some salamanders and some caecilians, which are a weird looking fossorial amphibian. But that's like, but the old school approach to studying this stuff. We're still only looking at maybe 5 or 20 different DNA sequences, and if you pick the wrong one, you might come up with pick the wrong gene to sequence, for example, you might get the wrong phylogeny. So the new thing is to look at the whole genome and to try to infer phylogenies from that. And so things that people have started to do from a sort of bigger genomic perspective are the position of turtles with invertebrates. Birds, it turns out bird relationships are not very well resolved. I don't think bats they still don't know where bats fall within mammals. Last I heard it was most closely related to this thing called a tree shrew, but I actually probably should have checked before I gave the talk. But I believe it's still questionable, and certainly relationships among extant human populations are hard to figure out. Like really, that's what I'm working on now. It's quite difficult. But so as a graduate student, I did some work on turtles, and here we developed a new method. Instead of looking at 5 or 10 genes, we could actually sequence more than 1,000. And it's probably kind of small, but so if you use morphology to study the position of turtles, these are all reptiles. You could put turtles next to lizards and snakes and the tuatara. You could find that turtles are actually, sister to all other extant reptiles. So birds are considered a reptile. Dinosaurs would fall in here somewhere if they were not extinct. And actually, when I made this figure, I didn't know, but there's even another paper that puts turtles as sister to crocodiles and birds. So if you just look at turtle bones, you can put them like wherever you want. The reason it's so difficult with turtles is they have a shell, like the plastron and the carapace, and they have a beak, and their morphology is like really specialized for aquatic life. And trying to figure out what the little hole in the head, matches the hole in the head in a lizard or a snake or a crocodile is not as easy as scientists would like it to be. Mm-hmm. So morphology didn't give a good answer. Early studies of mitochondrial and nuclear DNA typically put turtles with crocodilians and birds. But then just before we published our work, a new study came out looking sort of at a more genomic scale with these little tiny DNA sequences called microRNAs, which are, the reason they were interested in them was the idea is that once you gain a microRNA in your genome, you can't lose it. So there's less worry about homoplasy with the microRNAs. And that put turtles with lizards and snakes. So when we published our work, we found actually the turtles are back with what other people had already discovered, but with a lot more support. So these little values here in the phylogeny indicate, the closer you are to either 1 or 100, the more belief you have in that the data supports that topology. So apologize for all the black and white images. And then we applied the same approach to birds, and here it didn't work very well, which is kind of cool. So all the I flipped this around to make it fit, but the species pairs are pretty well supported, but back when you get to sort of the ancestors of many of the bird lineages, it's not very well resolved. It just looks like a big comb, and there's no bifurcation there. So that's even with like 1,500, I forget, it's like 500,000 base pairs of sequence, maybe a bit less than that. It doesn't resolve for birds. And the reason for that is probably twofold, but mostly because birds speciated really rapidly. So once they evolved this ability to fly, they could fly everywhere and find a new ecological niche and just take it over, and without enough time for evolution to leave a little fingerprint in their genome about this is what, where I started from. And the other possibility is that the sequences data that we have is a little bit short. I won't go into details, but it's most likely because they are rapid radiation. So this is something people are still working on. When you have or maybe not working on it, just may not be able to resolve it. That if you have a really rapid radiation of species, then they just don't leave enough information in the genome to delineate the evolutionary tree. And if you pay attention in the news, I think there's going to be more coming out about this with completely sequenced bird genomes, but it's not available yet. So, but yeah, I'm sure it'll be in the New York Times, although I'm not an author on the paper. Anyway, so human demography is another one that's pretty cool. So this is work in the lab I've just joined, and here, so now we're not looking at species, rather we're looking at human populations in Africa. As you can see, it looks like a mess. But so each little circle here, each pie chart is a different population, and then the proportions in here are showing the proportion of different ancestral groups that make up that population. And sort of the way this works is you plug your genetic data into a software package. You say, I think that there are 10 groups that were produced this genetic data, partition it for me, and then you can make these plots. So like over here in West Africa, all these orange pie, big pieces of orange pie are probably Bantu ancestry. So Bantu populations, or Bantu people, are agriculturalists, and once they developed the ability to farm, they could rapidly outcompete the other African people and moved all around Africa quite quickly and proceeded to transfer bits of their genome all over the place. So starting in West Africa, moving to Central Africa, and then probably down into even more Central Africa. Some of the cool stuff, so this big green piece of the pie are pygmy populations in Cameroon. There's also more pygmies over here. And similarly, the Khoisan people down in South Africa are really different from the other African populations. But as you can see, none of these populations here actually are totally, are totally unique, right? So they all have admixture with their neighboring groups, and that's what makes it really difficult to work on humans, because there's so much gene flow between populations.
Can I ask a question?
Oh, absolutely.
What, the fuchsia colored, it's too far away from me to see. So
like
see,
over here?
The fuchsia pieces, yeah, where are they?
I don't actually know.
Oh,
It's okay.
So then my other question is, so you've located, so that's the Bantu tribe. So
Yep.
what does that actually mean what does that equate to as far as to look at, these different populations? You may see some human differences. I mean, you can see in birds, beaks are different. So what does that actually mean, like Bantu? What does that equate to at the genetic level? Like a gene sequence that's different from the fuchsia, like what?
So it's like, when when people reproduce, their genomes get admixed.
Sure.
So it could be different chunks along the genome. So it's not like, I don't know if,
So as simple as like something like, blood type is not, blood type is, there's not that that many, but say an HLA type or something like that, a little bit more specific in areas, you can trace that back to like European ancestry or things like that.
no, if there's enough recombination between people, it just gets totally mixed up. But what's like cool, like in the pygmies, for example, there's little parts of their genome that seem to be less admixed. And those look like they fall on parts of the genome that have genes related to height. So, does that help?
Yeah,
sort of.
Yeah.
But the proportion is like randomly distributed for the most part on the genome. And this is actually not even in genomes anymore. This is like pre-genome data. So humans, people like, at least I'm interested in it. Presumably high school students like to hear about humans. So I've got some in there, but I'm not, I've only been working on it for 6 months, so I'm not quite as good. But also medical genomics and epidemiology are pretty interesting. There's a lot of work now doing people using cancer as sort of an evolutionary model. So you could take like a biopsy when it's first discovered, and then again 6 weeks later, and then again 6 weeks after that, and then study the genetic changes in the cancer cells, and they actually compete with each other, and some become more abundant and some become less. So understanding what makes some cancers, but you can use it to figure out why some parts of the cancer are more metastatic, something like that. I'm not a cancer biologist, obviously, and I don't have any slides on it, but that's a popular one. And then there's lots of work, of course, in disease origin, so HIV, Ebola, and lice. And so this just came out, I think, a month or two ago. And here they sequenced, or they actually got a lot of this data from online, I believe, and probably sequenced some of their own, but they could identify specific types of HIV that are associated with different regions. So in the Americas, it turns out that HIV first came to Haiti, and then from Haiti into the Americas, then rapidly expanded all over the world. So this subtype B1 is actually the most abundant HIV subtype, but it first came to the United States. So this is worked out from sort of evolutionary approaches. In Africa, they have their own subtype C2 that's unique to African populations. I think this is Southeast Africa, but I didn't check. And then the main thing, thrust of the paper, was they figured out where HIV originated. So it turns out it was in Kinshasa in the Democratic Republic of Congo. And they could they could determine it's probably 1920-ish when it was first passed into human populations, probably earlier than that, but that's when they have the earliest samples. And they could trace that it follows along these railroad tracks away from Kinshasa. And the kind of cool thing I thought was neat, there's like 2 lines in the paper, and they talk about how it turns out that men in Kinshasa in the 1920s were, on the whole, all circumcised. So if you've read some of those literature about how more circumcision in Africa should reduce the HIV, this may be some evidence that suggests that's not as crucial. Which, whenever you talk about body parts of high school students, they perk up. So, as well as
You're absolutely right. Yeah.
as well as undergraduates that I've talked about too. And then Ebola, of course, is all in the news right now. So this was also published a few months, or like last month. And this is out of the Sibeti lab at Harvard and the Broad Institute. And they got samples sent back to them sort of right as the outbreak occurred. And there's a really nice article, which I should have put a link to in here, in the New Yorker, like last week, all about this, written by the guy who wrote The Hot Zone. So Richard Preston, yeah. Not his brother Michael Preston. That's the thriller author. Which talks all about this. But the shorthand version here is they got the genomes back. They sequenced them rapidly, like in a month or two. They've got a paper in Science already. They can identify 50 unique little genetic variants that actually change this gene sequences in the new Ebola genomes. And they've made a little phylogeny, and they can see that it came originally from a strain that was first identified in, or was most closely related to a strain from the Democratic Republic of Congo, just like the HIV. And what was something else I was going to say? Oh, and so the really sad part of the story is that this paper's been published, then like 5 or 6 of the authors from Africa on the paper have since died from Ebola. So that's what the article is about in the New Yorker, if you look it up. And I'll try to remember to send it to Kristen and pass that on. And then, of course, again, evolution of lice is quite interesting, and it turns out, so these are mammals on this side, and then the parasite on that side, and rodents have their own unique louse, and old world monkeys have their unique louse, and gorillas have a unique louse. Humans have 2 different ones, and then chimpanzees have a unique louse. So it looks like, if you follow the phylogeny here, this louse here was the one that inhabits humans' hair, and it's probably what they had ancestrally. And then they got another one from gorillas, which is actually the crab. So in your pubic region, if you get lice there, that's from gorillas. And I'm not going to hypothesize how that might have occurred. But again, I'm sure that your students would be quite interested in discussing that. And so this is sort of the end of the phylogenomics part of the talk. And as I said, I didn't even really talk about inferring phylogenies from complete genome sequences, because there actually hasn't been that many examples of that yet. So that's like the cutting edge of things. And let's see. Okay, so some work I have done, which isn't really published. If you measure phylogenies all along a genome, like you line up, entire, multiple gigabase genome with similar ones, they actually change a lot as you slide along it. And again, it's because of this recombination. So figuring out ways of inferring what that means, I think is going to be really interesting. And again, it's all like super computationally difficult to work with. It's not like you can plug it into your computer on your desktop. You have to send it off to a computer cluster. So it's like that the big data thing is reaching biology for sure. Okay, so moving on to evolution of adaptive traits. There's lots of, I'll show like 5 examples of sort of the same study you'll see. There's lots of work trying to understand what makes a species and sort of genomically. So how many genes, what types of genes, where they locate in the genome, and then this other final question of sort of understanding, when you, so say you have 2 populations and they now inhabit different environments, when they become new species, is that because they've evolved actually new genes de novo, like separate mutations have occurred in different populations that make them different? Or is it because when they split off, they shared a lot of genetic variation, right? If you've got hundreds of thousands of individuals, there's going to be variation already there. And is that the source of the genetic differences? Does that make sense? So that's something people are doing a lot of work on, and it's not really clear which source of mutation or of variation is the one that creates species. But so the basic idea, if you compare populations that are recently diverged, like butterflies in Costa Rica is one of the examples I'll show you, most of their genome is identical because they're either recently diverged or they still exchange genes because they hybridize with each other, except for a few genomic regions. And it's these divergent parts of the genome that are different between the populations that likely contain the differences that make them look phenotypically different. So this is like the question you had before about, so this is how you go investigate that sort of thing. But in humans, it's really hard because they aren't really all that different yet. So you don't see like really nice big peaks. So I'm sort of repeating myself. How much of the genome needs to diverge in order to create new species? And then what sorts of genes and genomic regions are contained within highly divergent parts of the genome. So some of the first work was done on stickleback fish. And sticklebacks have there's an oceanic species that's large and has a lot of body armor. They have like this sort of bony body armor that helps prevent predation. And then in many cases, they speciated into little riverine freshwater like rivers or lakes just off the ocean, and then they lose their plates and they get a lot smaller. And so you can, if this hasn't happened that long ago, you can compare the genomes of the 2 types and try to identify the regions that are different between them. And those would be the sorts of things that help you live in freshwater versus saltwater, or develop bony plates or not. And so if you remember my little spike, that's what it looks like. So here's chromosome 4 of the stickleback genome, and most, here it's all the same, there's no divergence, and then bam, you get these big spikes where they're really different. And it turns out with sticklebacks, there's about 242 different regions of the genome that are necessary to make the 2 different species look different from each other. And the big outlier contains this gene called EDA, which is also important in human dentition. And I think if I recall, it's the one that makes people of Asian ancestry different from Europeans. So it's one of the genes associated with that. And then the other thing, which it's going to be sort of confusing, but sometimes bits of the genome can get flipped around. You can get an inversion, and then that prevents recombination from occurring correctly. And so those parts of the genome often are highly divergent because they can't actually recombine. And so that would be like a good project for a high school student to work on, maybe. So explaining why that's important. And then finally, so when they actually went ahead and looked in all these regions and tried to classify the genes, they found that it's sort of, the cool thing is they found that it's actually not genes in there. It's mostly these parts of the genome that regulate genes elsewhere. So binding sites. How much do you guys know about? Probably not that much about transcription factors, but it's where yeah, exactly. So But it's not the coding sequences that make the proteins, it's how the proteins are expressed. Those parts of the genome look like they're the ones that evolution is acting on. So that seems to be a repeated finding in many of these studies. So actually, I may as well say it now. So the reason people think that's the case, so if you have like a coding sequence that turns into a protein, like lysozyme in your saliva or something, if you get a, maybe that's not the best example, but if that was something that was really critical to your body and you get a big change in it, you could die right away. But if the change is just in how much is produced, it's like a gentler way of manipulating things. So that's the idea, is that if evolution's acting on the parts of the genome that just tweak it a little bit, then the consequences are less severe for the individuals. So they've done the same thing with butterflies. This is a really classic system. There's all sorts of butterfly diversity. They're really pretty, and they already know a ton about the genes that make the different colors. You can do cross the butterflies, like sort of animal husbandry kind of approaches, and figure out which parts of the genome contain the gene that produces different traits. But you can also do this sort of divergence approach. And this is actually, the data is messier. But all these, the region, so there's like 1, 2, 3, 4, 5, 6, 7, 8, 9 different regions that contain significant outliers. And they also happen to coincide with the part that they already knew has the wing color, the region that contains melanin patterning. So this, I think, if I recall, is whether or not you have these melanized bands on the wing, and the distribution of red patterning. I was actually an author on this paper, so I should know it better than I do, but I'm not really a butterfly biologist. I just wrote code. So they've done it with butterflies. Birds have been popular. I think this was just published this year. And so here they're comparing species of crows in Europe, and it's the carrion crow, which I forget which is which. One of them is all black, and one of them has gray patterning. And they can compare the genome again, and the top hit falls, this one here falls on a known gene involved in melanin pigmentation. So, and here you can see that instead of like 200, there's really only a couple different regions. So they've compared, if you go back, they have samples all across Europe. So that's, so when they compare the parts, the two birds that are most different from each other, they have the more of these outlier regions, but still it's not like 240. I think it's less than 10. So that's sort of for the really recently diverged species. It seems to be that's less than 10 regions of the genome are necessary to make pretty significant phenotypic or morphological changes, which is cool. Like, I would have thought it was a lot more. And they also seem to be clustered in different parts of the genome. Yeah, so and they've done the same thing with flycatchers in Europe. And again, I think they found one of the top hits has a melanin gene, and the flycatchers have, one species has got a paler plumage, and the other one's got a lot more melanized plumage. All right, so moving back to humans, the other thing people read a lot about, or you see a lot in the newspaper, is all these new agent DNA studies. And sort of the general protocol here seems to be find an old bone, extract the DNA from it, which you have to do really, really carefully, because if you contaminate it with modern human DNA, you're not going to be able to work out. Well, you can, but it's hard to work out which is which. And then you can sequence that DNA, sequence the genome, and then you can compare it to other genomes you have sequences for. And it's complicated, and it's like this multi-dimensional analysis, but they use this thing called PCA to flatten it down. And here, in this plot here, you have lots of samples from different human populations. And this purple guy here is this new genome from a 45,000-year-old modern human male from Siberia. And what turns out is he falls out like in between Central and North Asia. So instead of being like right in the middle of one of these groups of individuals. So like up here we have European populations, and then down over here you have African populations. It's got its own unique place on the map. So what the author suggests is that's actually because this lineage of humans went extinct. So that's kind of a cool thing to talk about. And then, of course, everybody's interested in Neanderthals. And there's been a lot of work sequencing Neanderthal genomes. There was a new one out in 2013. And so like what you can do in Africa, you can do with Neanderthals. And of course, so Neanderthals you already knew about. A few years ago, people found a bone in Siberia. Yeah, I guess it's like more like northern, I guess it's Siberia. And it turns out it's this whole new lineage of modern human they call it Denisovians. And they can actually infer the amount of ancestry that's Neanderthal. So Neanderthals contributed about 0.5% of their genome to Denisovians. Denisovians contributed 3 to 6% of their genome to modern humans. Neanderthals are contributing 1.5 to 2.1% of their genome to modern humans. And so we all have Denisovian and Neanderthal bits of genome in our genome inside of us. But the cool thing is sort of like that question about Africa is that our bits, like my bits, are different than your bits unless you happen to be like my sibling or something like that. So that because of all the recombination with our ancestors, that it's gotten all mixed up. But the proportions are probably pretty similar. They can actually go and predict, so that's not really quite true. They can predict which parts of the genome show the different bits of ancestry. So they can identify portions of the genome which are most likely to have come from Neanderthals. So if you're in a European individual, you can see there's a lot of regions. When you get down to East Asian individuals, I think the paper says it's a bit fewer, but I don't know, it's maybe a similar number, but certainly African people with lots of African ancestry have very little Neanderthal ancestry because this is out of Africa hypothesis, which really isn't a hypothesis anymore, but that Neanderthals came out of Africa and then didn't contribute anything back afterwards. And so, in conclusion, more data and genome sequencing is helping resolve fundamental questions in evolutionary biology. There's lots of work going on to work out phylogenetic relationships, evolution of adaptive traits, human evolution, demography. One of the new sources, actually, it's exciting because it's resolving questions about turtle evolution, for example. But we've got all these new loci under those adaptive peaks that nobody knows what they do. So we're actually going to have to go back and do sort of more traditional molecular functional assays and figure out what they're actually doing. And the methods for working with demographics in large genomes are a hot topic right now and really quite difficult. And then, of course, there's more and more data coming, so just finding ways to deal with that is really challenging. And then I snuck in, because of course I'm sure all of you have, at least when I teach undergrads, I dread the question that someone's going to ask me an idea question. So, you get the smartass in the back of the class says, I don't believe when we're talking about because the eye or the flagella or whatever is too complicated to have evolved without outside guidance. And sort of the safe answer, rather than getting into the specifics, is to say, just because science can't answer a particular question doesn't mean that the solution is to invoke an unknown creative force. And that scientists really are interested in these sorts of questions where they don't know the answer. And it sort of, once you start invoking things you can't explain, then you're not really doing science anymore. But not to get into the nitty-gritty of mechanisms of flagellar movement, that sort of thing. And then, of course, you could always point out that it doesn't make sense that a creative force would produce suboptimal designs, like extra parts of the wrist that pose as thumbs in pandas instead of just using the thumb, or goose flesh in humans that doesn't seem to do anything, or back pain because we have all these extra vertebrae down there that are not really ideal for a bipedal lifestyle. So now, of course, I'm going to get all sorts of questions about intelligent design, since I put that for last. Thank you, and thanks to